Hello everyone and welcome to this YouTube channel where hopefully I will help you teach the course of medicine through different subject and different chapter progressing one by one, and hopefully I will help you guide through it from a student's perspective. Now I know not everyone is preparing for USMLE or just is going to give USMLE immediately, so it is important to learn all the basic stuff first before you can advance to the more advanced level. So today the topic for us is chapter number 1, Gonad Development. Now before we progress even further, we need to ask ourselves, what is gonad? We need to know the definition before we even go into the chapter. So what is gonad? A gonad is a gland in which the sex cells are produced. So it would be sperm in males and ovum in females. Now that we know what the gonad is, we need to know where it is developed. So after fertilization, the gonad glands develop in the urogenital ridge, which is derived from the mesoderm. Now, during week 4 to 7, the gonads do not have a sex assigned. They are indifferent gonads. So we need to determine after fertilization whether the child will be a male or a female. And in this chapter, we will know how that is possible. Now, for the indifferent gonad to develop into either male or female, it needs the help of primordial germ cell, which is not available in the gonad yet. So where are those primordial germ cells? Those primordial germ cells are available on the lining of yolk sac. Hang on, let me pull up an image. As you can see here, the primordial germ cells are available on the lining of yolk sac. From here, they will travel all the way from back of the abdomen through, as you can see, the dark blue portion. That is where our indifferent gonads are. So the primordial germ cell from the yolk sac will travel from all the way over there to this dark blue spot in the urogenital ridge, where they will either determine if the gonad is male, which is testis, or female, which is ovary. Now we need to know how the gonad develop into either testis or an ovary. For that we only need to focus on two things: TDS and MIF. It may sound complicated, but it's really not. TDS simply stands for testis determining factor. It is self-explanatory. TDS, testis determining factor, helps determine the indifferent gonad into testis, and MIF is Mullerian inhibiting factor, which inhibits the production of ovary. Now let's go a little deeper so you can understand it. I can't just brush over this topic and just leave you wondering how it is formed and what happens. For the gonad to be a testis, it needs a Y chromosome. We don't have to dwell into the genetics that much. On the short arm of the Y chromosome, it has an SRY gene. What does that SRY gene do? It gives you the TDS, testis determining factor. Now TDS helps make testis, and testis produces Sertoli cell, which then produces the MIF, which is Mullerian inhibiting factor. Now this MIF is the key to not only develop testis. This MIF inhibits the production of ovary, which prevents the production of the female genital system and leaves the embryo with just one gonad system, which is testis. Now for the embryo to be a male, it needs at least one Y chromosome. It can have more than one, but if the embryo has at least one Y chromosome, then it automatically translates into a male. Let's just say, for an example, when there is a Y chromosome, it gives you SRY, which produces TDF. TDF then gives you testis, which produces Sertoli cell, and then which produces MIF. But let's say in a certain situation we have all that until the MIF. We have testis which produces Sertoli cell, but somehow if it does fail to produce the MIF, then the embryo will have both genital system, both the male and the female genital system. This is one kind of abnormality which you should keep in mind when you are reading this chapter. Now the second topic for this chapter is meiosis. Before we go any further, we need to know what meiosis is. You might have heard of mitosis, but meiosis may be new, so I will teach you the difference between mitosis and meiosis before we dwell any further. Now mitosis only involves one cell, which divides and produces two cells, whereas meiosis requires 2 cell division and then produces 4 different cells. Now, in mitosis, the daughter cell, the 2 daughter cell, they are genetically identical, whereas in meiosis, the 4 daughter cell are not genetically identical. So they do share their DNA and they mix up, which is meiosis, not mitosis. Now this is the most important diagram of this chapter. We will go through this diagram 3 times. I will explain each different thing each time we go through this diagram. Now the first time I will teach you how the meiosis 1 and meiosis 2 occurs. The second time we will learn the differences between the male and female, how it occurs differently in both male and female, and on the third time we will learn the time differences. When does meiosis 1 occur? When does meiosis 2 occur in both male and female? So let's go. So let's try the first one, how meiosis 1 and 2 occur. Now I taught you the difference between mitosis and meiosis. The type B spermatogonia, as you can see here, the spermatogonia and the oogonia, they differentiate by the process of mitosis until they are that differential that they shift to the process of meiosis, and that's when they are called primary spermatocyte and primary oocyte. Spermatocyte in the males and oocyte in the female. One important thing to notice here is that we have 46 chromosome and it is 2n, which is a diploid. Now during meiosis 1 we have DNA replication. 2 different cells, they mix with each other, share their DNA and produces 4 different gametes. Now during meiosis 1 DNA replication, as you can see here, before we had 46 chromosome, now we still have 46 chromosome, but instead of 2n, as you can see here, we now have 4n. So after replicating the DNA, we go through the process of synapses, crossover, and as you can notice the difference here, the dark blue one and the light blue one, they cross over, they mix their DNA and they produce 2 different secondary spermatocyte or secondary oocyte. As you can notice here, this one has 3/4 of its DNA and the other 1/4, as you can see here, the light blue is from the other one. And the same here, the 3/4 one is light blue, whereas the 1/4 is the dark blue, which is the proof that they mix their DNA while producing secondary spermatocyte and secondary oocyte for both male and female. Another thing to notice here, before we had 46 chromosome and 4, and now we have 23 chromosomes and 2, and this is important to notice. As a human being, a single person, either male or female, we only need 23 during our lives. When the process of fertilization happens, that is when we need the 46 chromosome. Otherwise a normal single human being have only 23 chromosomes. Now once we have the secondary spermatocyte and the secondary oocyte, the process of meiosis 2 helps them divide through the process of cell division, and which finally produces gamete as in either the sperm or the ovum. We produces 4 gametes, which is not the case in male and female. It is different for both, but we will go through it when we discuss it on the second time, the difference between male and female. So as you can see here, the process of meiosis through the cell division produces 4 gametes. Now it is 23 and 1n, which is called haploid. Before it was called a diploid, now it is a haploid. It has 23 chromosomes and a haploid, which is a testament that it is either a sperm or an ovum, a single cell. When it merges with another, either a sperm or an ovum, the different one from each other, then we have 23 plus 23, we have 46 chromosome and 2n. Then we are back to this. So after producing all of this, we are back to this. When either a sperm and an ovum meet, we are back to this chap again. Now going through this the second time, we will learn how it is different in males and female, how the sperm and ovum are different from each other, and how the process of meiosis differentiates between this. Now let's go through this again. We have 46, 2n diploid DNA replication, synapses, crossover, cell division is the same until this step, until the secondary spermatocyte and secondary oocyte. An important thing to notice here is, let's just assume the dark blue portion of this cell is the cytoplasm. What is the cytoplasm, you may ask? A cytoplasm in a cell is everything outside the nucleus. So whether we have the mitochondria, the Golgi bodies, the nucleosome and everything outside the nucleus, that is what we consider cytoplasm. So let's just assume in a cell the dark blue portion is the cytoplasm. Now as you can see here, the 3/4 part, the cytoplasm, the DNA, the 3/4 part, the retaining part is where the cytoplasm is. Let's just assume that. So in a female, the 3/4 part of a cytoplasm is what we will take and is what we will go through the process of meiosis to with. This, the one with only less cytoplasm, which only has a small amount of cytoplasm, this is what we call polar bodies in a female. It will be the same in male, whether it is this or this, they both are sperms and they both will go through the process of meiosis too. But in female, only this left side, the one with majority of the cytoplasm, that is the one which will go through the process of meiosis too, and the one with only a tiny amount of cytoplasm, that is the one which is called polar body, which will go on and die off soon enough because it does not have enough cytoplasm and only the female can give the child all the cytoplasm. So for the child to develop and for the child to have the mitochondria and produces the energy and everything, it needs the cytoplasm, which is only given from the mother and not the father. So important thing to keep in mind. Now again, in the process of meiosis 2, this, let's just assume the right side, this right branch does not exist in female. When we go through the process of meiosis 2, we go through the cell division and then we are produced with two gametes. Now the same thing, the majority of the cytoplasm is retained in one and just a minor amount of cytoplasm is given in the other cells. So again, the other one which will become a polar body will just die off and only one remaining gamete out of all four before, only one remaining gamete is what is left in the female. That is what our ovum is, our ovum or a egg. That is what goes through in the female and that is the only egg remaining from out of all four. It will be sperms in all four of them. It does not matter. The cytoplasm and that does not correlate in male. Will all four of them gametes will be sperms. But in females, only one of those gametes with the most cytoplasm is the one we will take. Everything else is just polar body and they will just die off. Now that we have learned the difference between both male and female, let's go through the time stems. When does process of meiosis 1 occur and when does the process of meiosis 2 occurs in both males and females? Now, the process of meiosis 1 in males only starts when a male hits puberty. Before that, spermatogonia are divided through the process of mitosis, and once they reach puberty, their spermatogonia will divide through the process of meiosis 1, which is primary spermatocyte, and after puberty is when the process of meiosis 1 starts in male. But in female, compared to the male, the process of meiosis 1 starts even before they are born. Just 5 months into the fertilization, just 5 months into an embryo, the process of meiosis 1 starts in a female. So the primary oocyte, they just starts developing straight into the 5th month of embryo. So now that we have process of meiosis 1, in males it is frozen into the first step until they hit puberty, but in female, one of each primary oocyte will go through the process of synapses each month. They will go through the process of synapses, crossover, cell division and secondary oocyte, and then they are arrested in that phase 2. Now in males, once the process of meiosis 1 has started, it will just go through the process of meiosis 1 which continues to meiosis 2 after they have hit puberty up until their death. So in males it is really simple. They just go through the process of meiosis 1, then 2, they produces 4 gametes which are sperms, and that's it. It's done. But in females, the primary oocyte just goes through the process of synapses, crossover one of them each month, and they are in the stage secondary oocyte, and they are arrested there. Each month they are arrested in the stage of secondary oocyte. They will not progress further to the process of meiosis 2 until and unless they are fertilized. Until and unless they are mixed with a sperm, they will not go through the process of meiosis 2, and if they are not fertilized, they will just fall off each month, which we call just periods in female. Now once it is fertilized, the secondary oocyte is finally allowed to go through the process of meiosis 2, where it will follow the cell division and everything and produce a gamete, which is an embryo. So in female, meiosis 2 is completed only and only if the secondary oocyte is fertilized. That is all from chapter 1, the development of gonad. Hopefully that was helpful. Hopefully I taught it from a student's perspective, went in all depth, and hopefully I was able to solve the basic problems and all the concepts for you. Let me know if I can improve anything, or if you want me to explain it even more depth. Hopefully this was helpful. This is my channel. Hopefully I will upload more videos and more chapters and not just slack off in the coming weeks, and that should be it. Hopefully you guys enjoyed. Like, subscribe, and I will see you whenever that is chapter number 2. I will see you then. Goodbye.